This is a presentation on the tendon fibroblast by Adam Brough, Cormac Clark and Jordan Condon. Firstly, we looked at what a tendon fibroblast was. A tendon fibroblast is an elongated fibroblast cell. It has numerous functions. Fibroblasts can condense a hydrated collagen lattice to a tissue-like structure. The rate of this is regulated by varying the protein content of the lattice, the cell number, or the concentration of an inhibitor. Tendon cells form a connective epithelial layer between the muscle and the shell in mollusks. When we looked at its location, we discovered that it is dispersed in connective tissues throughout the body. The tendon fibroblasts are originally derived from primitive mesenchyme, and in the lung they reside in highly complex multicellular environments usually opposed to the epithelium or endothelium. With regards to the shape, tendon fibroblasts are large, flat, elongated, triangular, spindle-shaped cells. They are 10 to 15 microns in size. Every 1,000 micron equals a millimeter. Their lateral edges are much thicker, smoother, and usually slightly concave. A tendon fibroblast has no detectable internal skeleton that by its rigid structure maintains the characteristic shape. Interesting facts. There are many different aspects and characteristics to a tendon fibroblast cell. Work in animals has shown that this tendon has the capacity for intrinsic repair. This process involves migration and replication. It has raised the question about the possibility that fibroblasts could be isolated from adult human tendons and maintained in vitro. Tendon healing will occur by adhesion. Primary intrinsic healing can occur and is indeed more advantageous. This is of interest when considering the possible effects of minor repetitive trauma rather than gross trauma of tendon severing.
In our study, we found that this cell comes in various shapes and sizes, as well as activated and non-activated forms can exist in less active states as fibrocytes. It is responsible for making the extracellular matrix. They are the easiest cells to grow in a culture, what makes them a favourite for biological studies. They play a role in wound healing following tissue injury. Fibroblasts move to the site of the damage, where they deposit new collagen and help in the healing process. Fibroblasts also produce glycoproteins and polysaccharides for the ground substances, a gel-like material that surrounds collagen fibers with dense connective tissue, which contributes to the structural integrity of ligaments and tendons and determines the physical properties of connective tissue. Many tendon fibroblasts are needed to remodel the tendon matrix by producing extracellular proteins under mechanical loading conditions. Therefore, to better understand tendon mechanobiology, it is important to determine the effect of mechanical loading on proliferation and collagen production of tendon fibroblasts. Cyclic stretching of tendon fibroblasts slightly increases cell proliferation but increases mRNA protein levels of collagen type 1. And here we have all our references that we used throughout our study. Thank you for listening, and we hope you enjoyed.